Ladies and gentlemen, thank you for standing by, and welcome to the Novavax First Quarter 2020 Financial Results Conference Call. At this time, all participants are on the listen-only mode. After the speaker presentation, there will be a question-and-answer session. To ask a question during the session, you will need to press star 1 on your telephone. Please be advised that today's conference is being recorded. If you require any further assistance, please press star 0. It is now my pleasure to introduce Associate Director of IR/PR, Erck.
Thank you, Operator. Good afternoon, and thank you to everyone who has joined today's call to discuss our first quarter 2020 operational highlights and financial results. A press release announcing earnings is currently available on our website at novavax.com, and an audio archive of this conference call will be available on our website later today. Joining me today are Stan Erck, President and CEO, John Trizzino, Chief Business Officer and Chief Financial Officer. Dr. Gregory Glenn, President of Research and Development, will be available for our Q&A portion of the call. Before we begin with prepared remarks, I need to remind you that we will be making forward-looking statements during this teleconference that could include financial, clinical, or commercial projections. Statements relating to future financial or business performance, conditions, or strategy, and other financial and business-related matters, including expectations regarding revenue, operating expenses, cash usage in clinical development, and anticipated milestones, are forward-looking statements within the meaning of the Private Securities Litigation Reform Act. Novavax cautions that these forward-looking statements are subject to numerous assumptions, risks, and uncertainties, which change over time. I'll now hand it over to Stan to get the call started.
Thanks, Erica, and thanks to all of you listening to this call. First and foremost, given the ongoing COVID-19 pandemic, I sincerely hope everyone on the call and all of your respective families are well and safe. I'll start the call today by discussing the historic funding from CEPI for our coronavirus vaccine candidate that we just announced a few minutes ago, and provide a brief status on our activities around Novavax COVID-2373. I'll then provide a recap of the positive phase 3 results for NanoFlu that we announced back in March, following a quick update on our ResVax program. John Trizzino will then provide a financial overview. After that, Greg Glenn, John, and I will be available for questions. As a prelude to my remarks, I just want to say that our accomplishments so far in 2020, including our progress in our influenza and COVID-19 vaccine programs, are the most impressive in the company's history. So let's get started. Beginning with our coronavirus program, we are thrilled that CEPI has chosen to invest up to $388 million of funding in support of 2373. As CEPI's CEO indicated, this arrangement represents CEPI's single biggest investment to date. This CEPI funding will allow us to advance our candidate through phase 2. It will also support the rapid scale-up of the vaccine antigen and our proprietary Matrix-M adjuvant, the critical components of 2373. In addition, this funding will allow us to dramatically increase our large-scale manufacturing capabilities. As we previously announced, we identified 2373, which will incorporate Matrix-M to enhance our immune response and immediately begin testing in preclinical studies. Our studies have shown that 2373 is highly immunogenic in animal models, including mice and non-human primates. High levels of spike protein-specific antibodies with ACE2 human receptor binding domain blocking activity and SARS-CoV-2 wild-type virus neutralizing antibodies were observed after a single immunization. In addition, the already high microneutralization titers seen after one dose increased 8-fold with a second dose. It is generally accepted that high titer microneutralizing antibodies are evidence that a vaccine is likely to be protective in humans. We are now launching our phase 1/2 clinical trial of 2373. In a few days, we will initiate a placebo-controlled, observer-blinded phase 1 portion of the clinical trial in approximately 130 healthy adults in Australia. Key assessments include dosage amount and number of vaccinations. The phase 2 portion of the trial will likely begin in Australia and the U.S. Importantly, we are just a couple months away from human clinical data, as immunogenicity and safety results from the phase 1 are expected in July. As I mentioned earlier, the additional funding from CEPI will have a significant impact on our ability to quickly begin manufacturing. It will now be possible for us to begin manufacturing the vaccine with a target of manufacturing 100 million doses by the end of 2020 and with a goal of escalating production to 1 billion doses in 2021. I should point out that these production goals are based upon two key assumptions. Those assumptions are regarding the actual dose of the approved product and the manufacturing yields that we actually achieve at large scale. We previously announced an agreement with Emergent Biosolutions to provide contract development and manufacturing services, including supplying Novavax with GMP vaccine product for phase 1 and phase 2 clinical trials. We are also exploring larger scale manufacturing capabilities globally. Details of those efforts will be forthcoming in the coming months. The significance of this funding cannot go understated, nor can our appreciation for CEPI's vote of confidence in our technology platform and progress. I'd also like to reiterate my thanks to everyone at Novavax. It has been an enormous undertaking to get this program up and running since January. But we have made immense progress in a very short period of time to make this vaccine a reality. Now let's move on to Novavax's most advanced program, NanoFlu, our recombinant quadrivalent seasonal influenza vaccine. While COVID-19 has grabbed the world's attention, we believe that NanoFlu will also be a game changer for the prevention of influenza. The phase 3 data we delivered validates its potential. Given our detailed March update for the program upon announcement of the phase 3 results, I'll just provide a high-level recap today. To put it simply, we achieved every single one of our goals in the phase 3 trial. The trial included 2,650 clinically stable adults, 65 years of age and older, at 19 U.S. sites. It was randomized 1-to-1 between NanoFlu and Fluzone, and subjects were given a single vaccination. The data we presented to date has been the 28-day results. The trial's primary objectives were to demonstrate non-inferior immunogenicity of NanoFlu compared to Fluzone quadrivalent using the day 28 ratio of geometric mean titers and the differences in seroconversion rates. Our primary objectives also included the overall safety of NanoFlu. We measured immunogenicity by HAI assays using egg-derived reagents. We achieved the primary endpoints for all strains included in the vaccine. NanoFlu was well tolerated and had a safety profile comparable to Fluzone quadrivalent. The secondary endpoints were to evaluate the immunogenicity using both egg-propagated virus and wild-type reagents for all 4 vaccine homologous strains and select drifted strains at day 28. For these endpoints, NanoFlu demonstrated significantly higher geometric mean titers and seroconversion rates than Fluzone quadrivalent across all 4 strains included in the vaccine, and NanoFlu also demonstrated significantly higher geometric mean titers and seroconversion rates than Fluzone quadrivalent for 4 drifted H3N2 strains not included in the vaccine but circulating this year. We believe this is validation of NanoFlu's ability to overcome issues related to egg adaptation and antigenic drift, which are major issues with current vaccines. We look forward to providing additional detail from the study in the near future, including the cell-mediated immunity responses, the T-cell responses, and microneutralization data. We believe these data will differentiate NanoFlu from leading licensed vaccines. NanoFlu has fast-track status from the FDA, and we plan to use the agency's accelerated approval pathway. We will conduct our required CMC activities in parallel with compiling the immunogenicity and safety portion of our BLA. We will communicate additional timelines once we have finalized our plan. Before I turn the call over to John, a quick update on our ResVax vaccine. We continue to believe in our ResVax vaccine. We're the only company to have demonstrated potent efficacy in clinical trials in both the older adult population and by vaccinating pregnant women to protect their infants. While not meeting the pre-specified primary endpoints for the phase 3 trials, we observed that the vaccine had significant effects on hospitalization and pneumonia in both trials, which provides critical insights on the continued need for our RSV vaccine. We believe that we can design an affordable pathway to a licensed product over the coming years and are designing new clinical trials that we believe could take us to a licensed product. Now that we have a strengthened balance sheet, we will continue to invest in this program. Now, let me turn the call over to John.
Thanks, Dan. Today we announced financial results for the first quarter of 2020. For the first quarter, we reported a net loss of $25.9 million, or 58 cents per share, compared to a net loss of $43.2 million, or $2.11 per share in the first quarter of 2018. The reduction in net loss was mainly due to reduced R&D expenses. Revenue in the quarter decreased 15% to $3.4 million from $4 million for the same period in 2018, and the decrease was primarily due to the completion of the PREPARE trial in 2019, partially offset by revenue recorded from CEPI's funding. R&D expenses decreased 52% to $16.9 million in the first quarter of 2020, compared to $35.5 million in the same period in 2018. This decrease was primarily due to decreased development activities of NVAX, lower employee-related costs, and other cost savings due to the Catalent transaction in 2018. G&A expenses increased coming in at $9.4 million in the first quarter of 2020 as compared to $8.7 million for the same period in 2019. As of March 31st, 2020, Novavax had $244.7 million in cash equivalents, and restricted cash. Net cash used in operations for the first quarter of 2020 was $23.1 million compared to $50.6 million for the same period in 2019. During this quarter, we further strengthened our balance sheet. In the first quarter of 2020, we raised $186 million in net proceeds, and from April 1st to May 8th, we raised an additional $74 million in net proceeds for a total of $260 million since the end of 2019 through our ATM offerings. Novavax's cash position as of today is now in excess of $300 million. That concludes my financial review, and now I'll turn the call back to Stan.
Thanks, John. We look forward to updating you on the phase 1 clinical trial of 2373 in July. And with that, I'll turn it back over to the operator for Q&A.
Thank you. As a reminder, ladies and gentlemen, to ask a question, you will need to press star 1 on your telephone. To withdraw your question, press the pound key. Please stand by while we compile the Q&A roster. And I'm showing our first question comes from the line of Michael Higgins with Ladenburg Thalmann.
Thanks, operator. Hi, guys. How are you?
Good.
Great. We're great. Thank you.
Congrats with the CEPI funding. That's impressive, especially to have the biggest they've put out so far. Looking ahead a bit, trying to understand with this ACT accelerator what kinds of margins you can expect. And do we look at anything that we're learning from Gilead's remdesivir and the discussions they're having with the investment community? Thanks.
Yeah, it's a bit early to predict margins at this point. We do know that we expect to be producing at very large scale, and we should be able to have efficient production. We don't know the cost of that production yet, and nor do we know what the selling price is. So it's a new world out there in a pandemic setting, and my expectation is that we'll have reasonable gross profit operating margins.
Thanks. The question would be looking ahead to this summer, just a follow-up from 2 months ago in the last quarterly call, which is once you've got data in July, you talked a bit about the schematics of it. You can give us kind of an update as to how you rolled into a larger study coming into the fall. Does the IND stay open? Is it a separate study, separate sites? Obviously, you want to review the data before you can start moving forward, but it sounds like it's happening in a bit of a rolling fashion. If you can give us kind of an update on that. Thanks.
Greg?
I can jump in. Yeah, so it will be done under IND. The data we filed to IND, and when we see the phase 1 data, we've written the protocol so that we can pivot rapidly into a later stage trial. Right now, we anticipate that will be a phase 2 trial on the order of 2,000 subjects. However, there's a lot of discussion about how one might accelerate that. So we're doing our best just to be prepared with sufficient animal data and human immunogenicity data to take the next step. And you may have seen we announced that on Wednesday we're going to give a more detailed update on the COVID preclinical and next steps.
Yeah, I noticed that. That's helpful.
Okay, good. I'm glad you saw that. Yep.
And then last one, I'm sure it's a busy queue. Just trying to get a sense for the global manufacturing. This is a heck of an update from the up to 10 million per month by year end to 100 million by year end, 20 possibly billions next year. I would think it depends not just on the yield that you mentioned on the call, but also securing access to manufacturers. Obviously, CEPI can help with that. But a lot of the folks that are involved with this, say the top 5 globally, they've had a lot of press releases and comments out about securing other manufacturers. How confident can you be that you'll have enough ability to manufacture? Is that something that CEPI helps with, that WHO helps with? How do we kind of think about that?
So we didn't just start thinking about that, and so we've been working on it for a while. We have been very optimistic about our data. We've seen this before. We've seen it with 2 coronaviruses. We've seen it with pandemic flu viruses. We've seen it with Ebola, where we get early preclinical data, and that in mice that translates into non-human primates, that translates into humans, and then phase 1 and phase 2 trials with immunogenicity. We bet, and of course, as you know, we don't have phase 1 results yet. We bet a while ago that our technology would give us good data in phase 1. So we didn't wait. We didn't lose 6 months by waiting for phase 1 data. We started on it right away and are securing sufficient capacity to be able to make the statement that we made in this press release. So,
appreciate it. Congrats again. Thanks, guys.
Mm-hmm.
Thank you.
Thank you. And our next question comes from the line of Mayank Mamtani with B. Riley. Pardon me, Mayank Mamtani, please check your mute button. Okay, and our next question comes from the line of Vernon Bernardino with H.C. Wainwright.
Hi, guys. Thanks for taking my question, and congrats on this amazing achievement. I'm very curious, and that's because I'm very excited for your very large amount of funding that you got from CEPI. I was wondering if you could tell me what exactly excited them the most as far as the vaccine programs out there to choose your program in particular. And as far as the candidates concerned, you chose CoV2373. How many candidates did you screen, and what led to the choice of that particular candidate for going forward into clinical trials?
Good. Thanks, Vernon. I'll answer the first question and let Greg answer the second part of the question. I think what got them most interested in our program was the fact that we are an experienced emerging infectious disease vaccine company. Our platform has produced, as I mentioned in my last comment, has produced vaccines and shown not only good immune responses and safety, but efficacy in models, in animal models and non-human primate models We've done in Ebola. We probably had the best immune responses ever gotten from a vaccine with our matrix adjuvant vaccine. And so I think it's the experience of that combined with the fact that our process has been scalable. We've done it with flu, we've done it with RSV, and so we have a lot of confidence we can scale it. And I think when you ask me when looking across the field and how do they compare to others, I think there are a lot of other good technologies out there that just have not had the experience that we have, and so there's and both with immune responses and with scale, and so ours is probably represents a fairly safe bet, and the others are exciting, and time will tell.
Yeah, hi Vernon, thanks for your support.
Hey, Greg.
Just to jump on this question, I think the hallmark of our vaccine is its immunogenicity. It's very immunogenic, and we were able to construct some good assays to demonstrate that. And so I think that's good. That was important. And so as you asked, we actually, I think, had about 30 constructs we made in a short period of time. And we did evaluate them based on several factors. So stability, productivity, because we knew a lot of doses would be required, and immunogenicity. And I think, when we look at our immunogenicity, you may know this, on Wednesday, we're going to provide some detail. We'll have a half an hour talk on our COVID program. You'll see our preclinical data. I think when they saw that, it is very compelling that the vaccine should work and be protective. So, I think that combination, immunogenicity, productivity, stability of the construct, these are proteins that are inherently can be unstable, and their structure is really critical. And I think, it's a mature technology. We just completed a phase 3 trial, with the same kind of construct. It's a recombinant nanoparticle, matrix M. We had very good results there. So I think all those things, have come together at this moment. We're very excited and grateful that CEPI stepped up to support us at this magnitude.
Great. Two follow-ups on that that are related. So part of the deal is to increase production of matrix adjuvant. What exactly is that for? And then, Stan, you mentioned that you saw high neutralizing antibodies just after a single dose. Is part of the thinking in all this such that if you need only a single dose, but your testing, let's say, leads you to 2 doses, but because of matrix M, the availability of it allows you to get away with a single dose, does that play into the whole thinking about the 1 billion doses by in 2021?
Well, I think I'll let Greg respond.
Yeah, I think we first of all, I think we're not seeing a real limitation with the adjuvant supply. So our staff has worked very hard to make sure when you look at those dose numbers that it's not going to be limited by either. So it's a relatively simple process to make matrix M. It's a very good adjuvant. We're going to look at this in the trial setting. As you note, after 1 dose, we get a very good response. We get neutralizing antibodies, and we see dose sparing. This is a virus, however, it's highly infectious. It has a very tight binding to the human cell. So disrupting that is, going to be very important. And there could be a lot of merits in having really high, neutralizing antibody. And we're going to weigh all those factors as we move into our late stage trials. We'll have the data to look at that, and I think we have assays that are really meaningful. They're conservative in terms of neutralization. They're functional in terms of blocking receptor binding. And we think it gets to the heart of the critical nature of how this infection spreads. So we're going to take into fact all those factors, I think, for making the dosing decisions going forward.
Great. Thanks for the question and the follow-up. So I'll get back in the queue with other questions or follow-up offline. Thank you. And congrats.
Thank you.
Thank you. And as a reminder, ladies and gentlemen, if you have a question, please press star 1 on your telephone. Once again, that's star 1. Our next question comes from the line of Mayank Mamtani with B. Riley.
Can you hear me guys now?
Yes, we can.
Okay, finally. I got disconnected first and then you couldn't hear me. Congrats on the update and thanks again for taking my questions. Very quickly on the phase 1 study design, I noticed that you are testing a dose without matrix M adjuvant. So I'm just curious, sort of what's the rationale for that, doing with and without matrix M, and also as you think about the IgG antibody assessment, just curious, how do you think about what you learn from there about the other endpoints, the more core endpoints of geometric mean, ELISA units, and others like CO conversion? Could you maybe talk to that?
Yep, that's a great question. So, look, whenever you do a trial with an adjuvant with a new compound like this, you need to clearly demonstrate the adjuvant effect. So, that really is important, and it allows you to compare and contrast the safety and immunogenicity. With our Ebola vaccine, which I think is a very good blueprint for the immune responses we'll see here, we could see the adjuvant was critical, and that was, important for going forward for regulators and, of course, public health and ourselves. So that's its role, is to demonstrate the need for the adjuvant. The different measures all. They are kind of, if you will, orthogonal looks at the immune response. The overarching response is a spike response, so we measure the IgG. Because people are immune naive, we expect to have very high levels of seroconversion on the order of 99%. So, that would be the norm here, and I think we're going to see that in our adjuvanted, especially our adjuvanted 2-dose formulation. The other assays, measure different things. So as I mentioned earlier, receptor binding is the first event. It's an important event. If you can block that, it's a very high affinity event. So it takes a really good antibody, in our opinion, to block that binding. Neutralization is a different look. It's probably a combination of what you get from receptor binding blocking and some other epitopes, some other immune responses to the rest of the spike protein. So, when we look at all of them. As you'll see on Wednesday when we talk about this, there's a really nice relationship between all those immune measures, which makes sense. And I think those different looks at the immune response really give us a lot of confidence that the vaccine should work.
Great, that's super helpful. And then on the Wednesday presentation, I'm assuming this is a combination of the non-human primate and the mice study that you've talked about. Any incremental color you could give on? I think you said 30 constructs you tested in mice, but in the non-human primate, was it just one candidate you tested? Or
Yes. We down selected. By the time we got to non-human primate, we had generated a lot of confidence in the construct we made. So the non-human primate is just the 2373 construct.
Okay, great. And this final question on NanoFlu, are there any alternative regulatory paths that become available, obviously given the current environment, beyond the ones you obviously have, like the fast track and the accelerated approval, that are potential options for NanoFlu?
That's an interesting question. So right now, no, I think we expect to go through accelerated approval. We have an obligation, to do a post-licensure efficacy study. I think we're on track for that. I think, it is very, very good data. I think, it's, intriguing. There was a lot of flu. When COVID came around, it was really dominating, and it's going to be interesting. I think, what COVID will do for flu is, we hope, generate interest in preventing this seasonal disease, because every year there's a large number of people that die from seasonal influenza, and we need a better vaccine. So I think we have very good evidence we have a better vaccine going forward, and so I think that's going to emphasize this COVID, this pandemic will emphasize the problem we see every year with an annual, seasonal influenza vaccine. And the issues we are addressing are the key issues: the drift, the fact that vaccines are not able to induce immune responses to recognize these drift strains, and especially around H3N2. So our data looks very strong in that arena. And I think, shortly we're going to be able to show the CMI data, and frankly, the phase 2 data will be published shortly as well. So you'll see that. We think that's the missing arm of a good immune response to flu in older adults. And I think you'll see our vaccine, I think we've mentioned it before, really gives a very good antigen-specific effector cell response. And we think that's an important piece of the puzzle here going forward.
Great. Appreciate you taking my question and look forward to both the nano flu update and then the July update on the phase 1 study for COVID-19.
Thank you.
Thank you. And our next question comes from the line of Vernon Bernardino with H.C. Wainwright.
Hi, guys. Thanks for taking my follow-up. One thing as far as the vaccine's concerned, you probably go into this at the World Vaccine presentation. What is the actual amount of protein for the COVID-2373 candidate that is actually being administered?
Yeah, so we're, I think we mentioned this before. We're using 2 dose levels in our phase 1 trial: 25 micrograms and 5 micrograms. It was our experience with Ebola, using similar levels, that we were really at the top of the dose response S curve. So 5 and 60 in that case micrograms were really no different in terms of immune responses. And so it's our expectation that's likely to be the case here. So obviously 5 micrograms is very nice dose sparing. 25 micrograms is relatively low, but so we're just doing those 2 arms with matrix. And then, as you mentioned early on, without an adjuvant as a control. So, that's our antigen dose level: 50 micrograms of matrix, 25 and 5 of the antigen, and we'll be giving that with 2 doses, or we also have one arm with a 1-dose immunization with 25 and 50 of matrix.
And as a follow-up, given sometimes the confusion of the symptoms as far as when you initially look at somebody who's infected with SARS-CoV-2 and people who are also infected with the flu, is there a potential to have actually a combination vaccine?
Well, yeah, I think, we think about that because our specialty is in respiratory vaccines, so it has that potential. I think we need to see where this will go. Many experts think this will become a common, annual seasonal issue, and it would make sense to have a combination vaccine. It's certainly theoretically possible. We could formulate the vaccine. It's the same platform. It's a nanoparticle. We're using matrix M in both those settings. That's a good thought.
Because if it totally doesn't go away, conceivably, if you added it to a seasonal flu vaccine, then you're kind of taking care of both, even and not really relying on the fact of the SARS-CoV-2 ever going away.
Yeah, it's a good thought. It's a good thought.
Well, congrats. I look forward to the additional data, and really rooting for you guys. So good luck and thanks.
Support. Yep.
Thanks, Vernon.
Thank you. And I'm showing no further questions at this time. I will now turn the call back over to CEO Stan Erck for any further remarks.
Yeah, thanks. As you can imagine, we're euphoric in the company. It takes a long time. We've been working to be at this point for at least the decade that I've been at the company. We've had, in the last 6 weeks, we've worked on a project that virtually everybody in respiratory vaccines have been trying to do, which is to get a better flu vaccine. Our goal was to get a differentiated flu vaccine, not be as good as the best flu vaccine, but differentiated from that, and we accomplished that. And that alone was a big deal. and then coronavirus comes along, and we do what we've been doing for the last half dozen emerging infectious diseases, and we have a lot of confidence, and we're being supported by large groups of academic and non-governmental organizations, and I think corporations that will help us get to the point where we can help solve this problem. So we're proud of the work we've done, and we're happy. We're finally getting recognized for it, and we look forward to reporting on data in a fairly short period of time. And thanks very much for listening in. And at that, I'll sign off.
Ladies and gentlemen, this concludes today's conference call. Thank you for participating, and you may now disconnect.